first thing that you absolutely have to get done, if you haven't already, is order the lab kit. And the second thing is you need to sign up for the library webinar. So that link opened up on September 7th. And you guys need to, if you have to have a library card that is current, and you get those each semester. So I posted instructions for getting a current library card, or you can go to the library website off of the college website and you can get a current library card. You have to have a library card before you can register for the webinar. But I would encourage you guys to get registered for that webinar because it's going to tell you how to do the assignment that's coming up. So that'll be coming probably after, not before the first exam, because I need to look at those dates again and see what her last date was for the webinar. I think it's sometime in October, but they're going to fill up. And unless they fill up, she won't open up more of them. And it's supposed to be something that all the Bio 1 students are doing. And I did it in the summer with my class. And so I'm expecting you guys to go to the webinar. If you don't, I have a very short abbreviated version of it that I can post as a video in Canvas, but it's not nearly as good as actually going to, it's kind of a backup emergency one. So I would make sure you have a library card and then use that library card to sign up for the, I don't know if they, I think she calls it the library information sessions. I forget what they're called, but you guys need to do that as soon as possible so that you're ready for when we get to that assignment, which, like I said, I'm not going to start that assignment until after the first exam. So probably next Wednesday, I'll start to talk about the assignment a little bit, and I'll check those dates to make sure that the assignment won't actually be due, obviously, until after all of you have been to the library webinars. Okay, and like I said, I'm not sure what her last date is. I think it's sometime in October, but I think it's like October, first, second week of October. So that'll be coming up. And so the instructions for that I put on the, they're on your Canvas. If I can find those. I think they're here in that first module. Yeah, no, I got too many things going on here. So this library card registration and the link for the information literacy assignment. So let's just check. So that assignment, obviously we can't do it until after October 14th. So, but I will start to be talking about it. Looks like the first one's next week. So after this first exam, but you have to register early, please. Otherwise, you'll be out of luck.
How do you sign up? Because I tried to, but I don't know.
So then you need to go to I would go to the library website then. I would go to the college website. And so this link didn't work for the signing up. This link right here didn't work for you?
No, because it was like expecting me to already know what my information was.
Right, you have to have, see how it says here, you have to have a library card. Step 1, get a library card. So you have to have your library card updated before you try to sign in and register for the meeting. So get your library card first and then click that link to sign up, because yes, it's going to ask you for some information from your library card.
Oh, okay.
All right, so today I wanted to ask, first of all, if you have any questions. Pretty much chapters 1 through 3 is about where you guys should be. 1, 2, 3, and that table from 4. And I would expect you to be working on chapter 5 this week. Chapter 5 is long and probably the hardest of the first 5 chapters. And then on Wednesday, I wanted to give you time to ask questions about chapter 5, because as I said earlier, the first exam is going to be next Tuesday, right? So, and then on Monday of next week, I'm going to move on and start talking about the cell. So questions you have from chapters 1 through 3 today. Anything about the chemistry, Angelina?
For chapter 3, are you want us to know like what kind of objects go into like certain pH scales? Like, are you gonna want us to know that battery acid is a number 1 on the pH scale?
No, that's not what's important from that. So to focus on that, so I believe I put in the PowerPoint set, and probably when I was talking about it in that video for chapter 3, there's a list of items, right, like examples of different pHs. That's not what's important. What's important is that you understand the pH scale. So you should have a definition of pH, definition of acids and bases. Have to remember that acids are proton donors to the solution and bases are proton acceptors, right? So we're kind of using, I think that's the Lewis acid-base model for those of you that have already had chemistry. So that's what's important to remember. It's important to remember how that pH scale works, that each step on that scale is the power of 10 because it's a log scale, right? Yeah. So in pH, we're actually measuring, I'm actually going to go to that because that's kind of important. Oh, so it's just in the video. There's no set of PowerPoints just for chapter 3. Okay, so let me try something else. Let's just go to this. Let's just look in the book. Get rid of my inbox, see what else I can get rid of up here. Yeah, it looks like everyone in the country is logging on to this right now. All right, let's see if I can get to chapter 3 here. And I believe it's the end of chapter 3 that we're looking for, is where the pH scale shows up. Oh, this would just be so much easier if we were all together in person.
It's 3.3.
3.3, all right, thanks. Yeah, it would be nice if I actually had a book too, but like I said, like I think I emailed you guys and told you, it's in my office, which I can't go to. So, and since they're expensive, they don't want to send me one either. Like, I'm just like, oh, could you send one to my house? And they're like, well, we could, but couldn't you go into your office? And I was like, so for pH, I'm trying to get to that picture and it's moving very slowly. So there should be definition, acids and bases, and I was kind of looking for that. Why can't I find it? So nice video for you to watch. I think it's part of one of your homework assignments. I'm not going to be asking you like these chemical names that the water molecules dissociate into. That's not important. This is what I was looking for, right? So this kind of sums it all up in one picture. So in a neutral solution, which is pH 7, and the reason it's pH 7 is because there's equal concentrations of protons or hydrogen ions and hydroxide ions in the solution, whatever it is. And technically it would be the only solution that would be the pH 7 would be pure water. This, you can't find this on Earth, right? Not even in Fiji. You have to actually manufacture pure water so that it is a perfectly neutral pH. These substances here, if you guys turn off your microphones because you're hearing me through someone's phone or through someone's computer. These substances here, these liquids that have a pH that means they're acids, means they can donate, that when you put them into a solution of pure water, they have excess hydrogen ions that they can donate to the solution. So yeah, there's more red plus hydrogens here than there are hydroxide ions, right? So there's more protons in this solution. That's a solution that's an acid has protons to donate to the solution. We can't measure these protons one by one because there's so many of them, there's billions of them. So we measure them electrically. That's when you put those electrodes into a solution to measure pH, right? So that to measure them, you have to measure them on a log scale. So a logarithmic scale of power of 10. So each step on this pH scale, if you go from 5 to 4, that's not one step on the pH scale, that's 10 times more acidic. And from coffee to vinegar is 100 times more acidic because you're going 2 steps on the pH scale. And the same for bases. Bases are usually defined as, we don't say they're OH or hydroxide donors, we say they're proton acceptors. So there's a lot of things in Bio 1 especially, or in biology and chemistry, that the things are opposites, and it gets tricky to remember both of them in the correct order. So what I always tell my students is just pick one of them and memorize that, and then remember that the other thing in the set is the opposite of that. So what I mean by that is I would remember pH, that's what it stands for, the potential of the hydrogen ion. So it's telling you in its name what it does. So you're measuring the electrical potential of how many protons there are, how much positive charge is in the solution. So acids are proton donors to the solution. Lots of protons gives us a low pH or a pH number less than 7. And that's what I would remember. And then if I ask you a question about a base, I wouldn't try to remember both of them. I would just say, okay, I know what an acid is. The base is the opposite. And then puzzle it out. You'll have plenty of time in the 50, 60 seconds, 45, 60 seconds to do that on a test if you actually know the definition of an acid.
So we don't need the calculations to find the substance or anything?
No, we're not calculating moles and stuff. Go to chemistry, learn all about it. That's more chemistry than we need, right? This is chemistry we need, right? That the P stands for the potential of the hydrogen ion. It's the inverse log of the concentration of the hydrogen ion, right? And it's a log scale that you have to know. And then probably even more important than that is the part that comes after this, I think, about buffers. Because in living systems, this is what's important to cells and to organisms. This is part of what we would say homeostasis, or part of those characteristics of living things we had the first day, right? Part of regulation. All cells have the ability to buffer changes in pH. If they don't, they'd be dead. Because every time a cell produces energy, it's making acids. So it has to have a way inside the cell or inside the organism to be able to moderate drastic pH changes. And that's what a buffer is. And then they give you this specific example because this is one of the most common buffers in biological solutions, right? It's carbonic acid. This specific example, do you have to know this? Am I going to ask you how to solve this particular chemical reaction? No, but they're going to ask you about it in anatomy and physiology when you get there, and they're going to expect that you know what a buffer is. So you do need to know the definition of a buffer, and you do need to be able to understand that a buffer moderates pH inside living systems, right? And it's usually, it's almost always a weak acid and it's salt, right? Those of you, again, those of you that have had a lot of chemistry, you'll remember that. No, and for focus, guys, whatever I assign in the homework, Like I sit and go through each of those homework questions. So the things that I've picked out of, and I know like this isn't important to you, but when I look in mastering for each chapter, like for chapter 3, there are over, I think it's 130 items that I could ask you and put in an assignment for homework or for a quiz. So I sit and look through each of those items and figure out, okay, is this something that I'm asking them to this depth of knowledge, or I just need to know them to know the definition, or I want them to watch the video about pH, or I want them to watch the video about, different things about water. So the items that are in the homework are chosen by me. They're not just random, oh, this is some professor in Idaho who gives his students what he gives them for chapter 3. I sit and look through each one of those 130 items and I pick out the ones that I know you're going to have to know and that I'm going to ask you about on the test. So the point of the homework is to help you focus on the information so that you can prepare. And once you've completed them, you can reopen them anytime and do them again for practice, right? So they're available for review. You can't change your score on them, but you can go through them again anytime you want. And it looks to me like, so I sent that email last night. I wanted to mention that as well. I sent that email last night about your scores in mastering before I really had looked closely. So my apologies again. Please don't send me another email that tells me your score. I can see them. If you can see them, I can see them, okay? The mastering gradebook puts the assignments that are active first. So when I looked yesterday afternoon, assignment 1, the homework for number 1 was already like in the 3rd position on the chart, if you can think of it that way, right? And so when I looked down and I saw that for homeworks 2 and 3, for chapters 2 and 3, there were some people who had finished them, but there were a lot of people who didn't have any score. I panicked and I was just like, what? Why can't I see this? So I tried to sync the grades real quickly and it took time. And then I tried to open my Canvas grades. And I was like, they're all sitting there in Canvas. What's going on? So it's a learning process for me. I've used this mastering program. I kid you not, my subscription just expired because I've been using it for 8 years, right? So I'm very familiar with it. They just keep changing it, especially now, especially to the 6th edition or the 12th edition with so many students using this completely online. They've made a lot of changes and improvements and they don't particularly tell us all of them. You just kind of find out when you sit down to use it. So all of you that finished homework number 1, I have your scores. I know what they are. If you can see them in Canvas, I can see them in Canvas, okay? So I'm not going to answer all those emails. Like I think there were 43 of them that were, this is my score. Can you see it? I just want to double check. I'm not going to answer all those emails. I just want to let you guys know. I'm telling you right now, I see all of them. If you can see your score in Canvas, I can see your score in Canvas. Okay. There are only, out of all 88 of you, there are 3 or 4 people. There are 2 people that have not, doesn't even look like are even in mastering yet. And so that's really what I was looking for last night. I wanted to make sure everyone was in and had done the homework for chapter 1. And most of you are on your way for chapters 2 and 3. Great, keep going. I think they're due today at midnight. So work through those. And then there's no homework for chapter 4, as we mentioned at the beginning, because all you need to know is that chart from chapter 4. And let's see, I think I actually, but I know if I close this, I'll regret it, but whatever. I'm just going to close it anyway. I think that, I think for chapter 4 in the module, there's just, I just put that, that table so you can just see it. So it's in module 2 and I think it's, maybe not. I thought I just put that one picture in for chapter 4. All right, I'll try to find that and stick it in there. I thought I had that just as a.
I have.
Sure. Yes, I can't see you right now, so sorry. Let me get out of some of these things.
So you mentioned that you pick which questions to put on the homework assignment.
Yes.
Is it the questions that say like pre-test that we should be studying?
No, all of them. All of them. So those are just different categories that the publisher gives them. So, and sometimes I arrange them so that the ones that are reading questions are first, and then the pre-test questions are first, second, and then the activities are in the middle, and then the ones that are more like test questions are at the end. Sometimes I arrange them that way, sometimes I don't bother. So no, anything that's in there, that's all, anything that's in the book is fair game for the test. It's just how it is. It's science, it's biology. You have to know, in reality, you have to know all of this at some point, right? The things that are in the videos and the note outlines and the things that I've picked out to put in the homework, those are the things that I think are the most important. So those are the things I'm going to ask questions about. I'm not using those questions. I've written my own questions. So those questions are to help you prepare for the exam, but I'm not going to ask you the exact same questions.
So we should know like the equations then of like the chemistry stuff, because it was asking us like how to calculate this equation and everything.
Right. In which chapter was that? Chapter 2?
Chapter 3.
And what was it asking you specifically to calculate?
I can't remember, but there was an equation that we had to do some type of math with, and confused because I think you said we didn't have to, but.
Well, if it's in the homework, then it's something that I think you should be able to figure out from reading in the book. And a lot of times those things that ask you to actually do the math are associated with a tutorial. So if you watch the tutorial and that's a question that comes after that tutorial, then yes, I would expect you to be able to do that.
Are you talking about the logs with pH?
I don't know specifically, like off the top of my head, I don't have those in front of me. Let's see.
I was asking Danny to see if he remembers.
Right. I don't think he remembers.
Yeah, because those ones, you kind of just hit the log button, put the number in, you're good.
Right. I don't think there's a lot of actual math that you have to do. Right.
Yeah, I found the specific question I think he's asking about. It's what is the pH of an aqueous solution with a hydronium ion concentration? And it gives the H3O plus equals 2 times 10 to negative 14 M.
And so it's asking you if that's an acid or a base, basically.
No, it's asking us to actually solve it. Like we had to put in a number for the answer.
Yeah.
I don't remember that that's in there, but I'm not going to ask you that on a test. I don't think. I would say I'm about 150% positive. You don't have to do a log calculation on the test. The point of including that in the homework is to help you understand that the pH scale is a log scale, right? So yes. And doesn't it give you a hint if you do it incorrectly the first time?
Yeah, it has the 4 different available hints.
Right. So you have to utilize those as you're going along in your homeworks, right? All right. Other questions from 1 through 3?
Hi. Could we go over hydrogen bonds, please?
Sure.
Yeah.
Sure. Let me so I'm on my computer. So hydrogen bonds, I believe, are in chapter 2. So if I'm just going to grab the chapter 2 PowerPoints here real fast if I can. Oh no, that's just the, dang it. All right, yeah, I'll just get a blank screen and do it. Stop sharing. Your view. All right, so you'll just have to walk over here with me. All right. I'm just trying to close some of these. Stop sharing there. And I will get rid of my fancy background here so you can just see me instead. Why can't I do that? Enable touch on my parent voice display, turn on background. There we go. None. Okay, so let me put some books up here and we can. I have a little box that works really well, but it's in our other room, our other. All right, I know there's some glare. I'll turn off this light. All right, so I know there's a lot of glare on the board, but if you guys can just bear with me. We'll see.
We can't see you.
Okay, sorry. Let me get, let me try to figure that out real fast. So let me start my video. There we are. Oh, can you see me? Yay. Most of the glare. Yeah. All right, so, yeah, but no. Different time of year. Okay, so hydrogen bonds real fast. I'm trying to make this so you guys can see better. I don't know what that is. Yeah, there we go. There. So it's bigger now, hopefully, right? Okay, so a hydrogen bond is tricky because it only happens when you have one molecule. So water is probably the best example, right? So I'm going to draw oxygen here with its 8 protons, 8 neutrons, and 8 electrons. So 2 electrons fit in that first shell, and 6, right? 1, 2, 3, 4, 5, 6 electrons in the second shell, make the total of 8, right? So this is oxygen. Then you're going to have 2 hydrogen atoms that are going to be covalently bonded to this oxygen. One sharing its single proton and its single electron. What's the matter? I know, you can't see it very well. I get it. It's the best I can do. Right, so can you guys, you can see that a little bit, right? So the idea, the important part is that when I get to the part about the hydrogen bond, I'll try to do it over here in this darker area so where you can see it a little bit better. But it's not a perfect system, but it's the best I can do, okay? So if you guys turn your microphones off so that every time you move something in your house, it doesn't jump to you, right? So 2 hydrogen atoms, each with 1 proton and 1 electron, because hydrogen only has 1 proton. Its atomic mass is 1 and its atomic number is 1. So the little dashed lines are the electrons. So this is a water molecule. Okay. So what a hydrogen bond is, H stands for hydrogen bond, is when one of these hydrogens or both of them are also attracted to another molecule, right? So a hydrogen bond is when a hydrogen atom that is covalently bound, and it's usually, we say, to an electronegative molecule. So it's usually an oxygen or a nitrogen. And it's also attracted to another electronegative molecule or another oxygen or nitrogen, right? Electronegative encompasses all the atoms that have electrons that can really easily steal electrons from someone else. So water is full of hydrogen bonds. Each water molecule can actually form 4 hydrogen bonds with neighboring water molecules. So that's what this means. So this hydrogen is officially, like if we were to draw this in a ball and stick way, we would go like this, right? We would put, because this is the oxygen, these are the hydrogens, they're officially bound, covalently bound together with a line. Hydrogen bonds are always dashed little sets of little dots because it's a weak bond. And it's this hydrogen, because its electron is spending most of its time around the oxygen, it's like, I have this positive charge, and so I'm kind of attracted to something else that's negatively charged. So I need to erase this guy and put another oxygen here. So a second water molecule that looks like this, there's a little bit of negative charge on this oxygen because most of these hydrogen's electrons are spending their time around oxygen. So this hydrogen is like, I'm officially bound here, but I'm also kind of attracted to you because you have a negative charge right there. That's the hydrogen bond right here, this dotted line. And each water molecule can form 4 of those because these electrons in the outer shell, there's 8 electrons, they're evenly spaced, zooming around there. And because the oxygen is so much more, has so many more positive charges than the poor hydrogen, right? So this would be, water is held together by a polar covalent bond, meaning the electrons from the hydrogen spend more of their time around oxygen than they spend around the hydrogen. These hydrogen bonds are usually established in polar covalent molecules. So you have to have like a difference of charge in order for that to work. I'm trying to make that a little bit better. I think it, piece of cardboard and put up in front. I don't know where that light is coming from that's so badly affecting us, but that's the best I can do. Hopefully it made it better, not worse. I'm asking that, I forgot.
Thank you.
You're welcome. So probably, I think there's a, that's probably in that video from chapter 2 or 3. Probably chapter 3 probably does hydrogen bonds again, because that's so important for water, right? Water, that it has those 4 hydrogen bonds, it gives it all its special properties, which is most of chapter 3, just the list of those properties and all the answer to all those questions. Why does water do this? Why can water do that? Why is water so good at this? The answer is always because water's polar covalent and water forms 4 hydrogen bonds with other water molecules. Like that's. Where that's coming from. All right, I'll try to work on that. We have to use the board. Maybe I'll try and move it up to my other room. All right, other questions, worries, concerns?
Can you go over orbitals and like the structure of molecules?
Yes, because then that would make this make a lot more sense. Sure. Let me grab all right, Kelly, I think I'm gonna switch to see if the whiteboard works better with all this glare. See if I can get it to work a little better. Maybe, maybe not. Maybe if I just write over here. All right, so the number of orbitals that you guys need to know. So let's start at the beginning, right? So the atomic number of an atom, which you would find in the periodic table, or on a test situation, I would have to give it to you, right? I wouldn't expect you to know where the periodic table was and how to use it. But the atomic number is equal to the number of protons that an atom has, and the number of protons is the number of positive charges that the atom has. The atomic mass, right? That's the number of protons plus the number of neutrons, and neutrons are just like they sound. They don't have a charge, they're neutral, but they weigh, they have mass, right? So the atomic mass is the number of protons plus the number of neutrons. We assign protons as a mass of 1, basically, and neutrons as a mass of 1. And then the electrons weigh, but they have mass, but they don't have very much mass. And the best example I can give you of that is if I compressed all the electrons in my whole body or your whole body, and I compressed just the electrons and weighed them, they would weigh a paperclip. So we don't count them in the atomic mass, okay, unless we're doing some serious scientific notation. The reason these numbers are important to you is because you do have to know, if I give you an example like oxygen, because that's something or let's do carbon. Right? So carbon's really important, and it's going to be super important in chapter 5. All of them are organic molecules. They all contain carbon. So we'll talk about carbon. So carbon has an atomic number of 6 and an atomic mass of 12. And that tells you all the things that you need to know about it for it to do its chemistry. 6, the atomic number of 6 means it has 6 protons. So if we were to draw a carbon atom, we would put 6 positive charges, 6 with a plus or 6 with a P and a plus, to indicate the positive protons inside the nucleus of this atom. Its atomic mass is 12, so 12 minus those 6 protons, the atomic mass minus the atomic number, tells us how many neutrons, which is 6 neutrons. So that's where the 12 is located. The protons and the neutrons, they're in the nucleus of the atom. Now, if this atom is neutral, meaning it's not, there's no plus or minus sign above it there to indicate that it has a charge. If it's neutral, that means it has the same number of electrons, which have negative charge. And we usually indicate those like that, E with a minus sign. Same number of electrons as protons. So we fill the energy shells, and 2 electrons fit in the first energy shell, and 8 fit in the second, but we don't have 8. Carbon only has 6 electrons, so we just put 2 there. So then we're going to put 4 in the next shell, like that. This outer shell, you'll also hear people call it the valence shell, because that's where the chemistry is going to take place. And this is what dictates whether carbon is going to be forming ionic bonds or covalent bonds. The first energy shell holds 2 electrons, the second shell holds 8 electrons, and it's not important that you know the shapes of the orbitals and the different subsets of orbitals. Don't care, go to chemistry, learn all about it. But you do need to know how many electrons fit in the first 3 shells. First shell 2, second shell 3, third shell also 8, sorry. First shell 2, second shell 8, third shell 8. We don't need to use the third shell. We don't even need to, we're not even filling the second shell in this case. Because carbon has 4 electrons in its valence shell or its outer shell, it's going to be very likely to share or form covalent bonds because it doesn't have enough positive charge to steal electrons from anybody else. And it doesn't have so few that's willing to just give them away. So if you watch the video from chapter 2, when we go through ionic bonds and covalent bonds, right? That's what that's all about. How many electrons are in that outer shell, and then what's going to happen to that atom as a consequence of that. Better, Colleen, or worse?
No, it's fine. I was just like asking that because a few of the questions were asking about like the orbitals, and then one was like, why is a nitrogen atom like structured the way it is? Like, or some, like there was a molecule, there was an atom in the middle, and then like 3 hydrogen atoms coming out in like a triangle shape, and it's like, why is it shaped like that?
Right, it's shaped like that just because the spaces of the electrons.
Okay.
Right, and how the hydrogens are oriented around that nucleus of nitrogen as negative. So it was NH3 probably, right? And hydrogen can also be it can also form ammonia, NH4, but when it's in ammonia state, it usually has a positive charge. It's like NH4 plus. So try to do the chemistry with moderation, right? So this isn't chemistry, but we need to know much of the chemistry that I put in the homeworks. We need to know that in order to be able to do the biology.
Okay, thank you.
Okay, so don't try not to obsess about it. But if I don't put any hard questions in there, then, and you take the test, then you'll just be like, well, we didn't know we had to know that, right? So it's difficult because there's way more things in this textbook than I can obviously ask you on a test, but there's a lot of it that's going to repeat and come up over and over and over again. I would, for those of you that are moving through this, right? So for I would finish your homeworks for chapters 2 and 3. I think those are due tonight at midnight. And then I believe the reading assignment is on for chapter 5 for this week and maybe chapter 6. Remember, those reading assignments are to just keep you on track, right? They have a due date on them. Otherwise, I can't put them in mastering. And I put them in just to make sure, just to remind you to be reading the chapters and which chapters to be reading and which parts of which chapters to be reading. So before I click the go button on the reading assignment, I do look through the chapter because occasionally, not very often, but occasionally there are some things that I'm like, not important, you don't have to know that. And then I will take that out of the reading assignment. So it is worth sitting there and going through the reading assignment, even if you don't like to read it online, if you'd rather read it in a real book, it's worth looking at it because it'll tell you which sections that are going to be included in the homeworks and then on the test, right? And those don't count for any points. So they're just for you to sort of stay on track, right? So those will always be, and I can't assign, if I assign them, then they show up in the grade book. So I can't, they'll just always be zeros or dashes, but they don't have any weight in your grade. So I've had a lot of emails about people saying the reading assignment says 0 and I read it. They always say 0, right? And they always say 0 even after you click the button because they don't carry any points in, I'm not forcing you to read them and count that toward your grade, okay? So a lot of emails about that. The library thing we already talked about. I can see that, like I said, I can see that most of you are in, almost everyone's in except 2 people don't have any scores for anything. So I'll try to contact them separately. And as I said in the email, for those of you that joined late, right, I think I put it in an announcement, right? So previously in the summer, when I would click on the button in the gradebook that said, contact all the students who have not submitted, only the people who hadn't submitted showed up. And this time it didn't work like that. So there's a delay always between mastering and Canvas. and I didn't take that into consideration. And so I had synced the grades, but I didn't wait like an hour and a half and then go back to see that they had gotten dumped into Canvas. So if you completed the assignment and you can see it in Canvas, I can see it in Canvas, right? If you completed the assignment and it's not in Canvas, then we have a problem. So those are the people that still need to talk to me, like during office hours or at the end of today. All right, anything else that's on your mind? Right, so can you see that, right? So this week, read chapter 5, right? And then do the homeworks. I put read chapter 6 on there for today because I want you to realize you have to start to kind of get even or a little bit ahead of me. There will be some math in this skills measurement thing here, and there's also probably going to be another measurement lab that is going to go in here, maybe this week. I don't know. Depends on when we get our kits. You guys got any word on that? When if you heard anything from the bookstore? Yeah, I guess it's only Monday, right? So probably not.
No, they're still saying back order.
They're still saying backward. All right. I think there might be a measurement exercise in the box. I'm not sure. I have to go back and check that. Yeah, it was supposed to be September 16th. That's what they told us originally. So the bookstore might be saying end of the month, thinking we'll have them and you'll have them in your hands by the end of the month. I don't know. I'll see if I get any update on that today. All right, other content questions from 1 through 3. Remember from chapter 4, it's just that one table that you need to know. And for that, like for example, and on Wednesday, since Wednesday you should have read chapter 5, I would watch those videos for chapter 5 because it goes through exactly what you need to know, the structures and the functions. And then Wednesday, I'll put up on the whiteboard, before you guys come to the Zoom, I'll put up some molecules and we can go through them and see if you can identify them. For chapter 5, you will have to be able to, on the quiz, you'll have to be able to identify the molecule and tell me its function. So you'll have to be able to identify not the name, like not specifically glucose, but you have to be able to say, oh, that's a carbohydrate, and this is what carbohydrates do in cells. So that's all of chapter 5, structure, function, structure, function, structure, function. And it's divided up into 2 videos because it's very long. And the note outline is good. It has everything and there's spaces for you to write on it. And the homework is pretty good for chapter 5 as well. So try to take a look at that before I see you on Wednesday. And for chapter 4, right, so they'll just be things like this. Oops, I'm doing it backwards. Summer. Let's try. All right, so if we have like this, phosphate, right? So like there will just be a picture like that on the test, and you're gonna have to be able to tell me. Let's get it out of shadow there. You can see more of my coat closet. Right, so this, you just have to be able to say that's a phosphate. Or for example, if it's like this. You need to be able to say that's methyl. It's methyl group, right? There are 8, I think, in that box. Carboxyl, carbonyl, methyl, ones that you're going to see over and over and over again. So for chapter 4. All right. So I will post this video that we just did going through this stuff, this specific stuff, but it's not all of it, right? So I would watch the videos from summer that are labeled summer, chapters 2, 1, 2, 3, and 5, because those are long lectures. Those are going through it just step by step with the note outline, what are the parts of that particular chapter that are going to be important. Sure, you can ask as many questions you want. No, Colleen, you're done. You only get 1 question a day.
So I don't know how important this is, but it kept messing me up on the chapter 3 homework. I don't understand the whole why does oil repel water thing, and why the amphithetic molecules line up at the surface of the water.
Oh, well, yeah, that's kind of important, right? So it all has to do with its charge, right? So the reason that oil and water form a barrier is because oil, predominantly fats, are non-polar. So when they make a molecule, so they're leading you. They're trying to get you to start to think about phospholipids and how they form membranes in cells. So it's kind of a foreshadowing event, right? So these molecules, this is a phospholipid. So this molecule has two parts to it. They're trying to also tell you that any molecule, even like water itself, can have areas where it's more polar, more negatively charged, and more positively charged. And when you make longer molecules or larger, more complex molecules like this phospholipid, this part, the phosphate head, has a negative charge, which we call hydrophilic, because it's going to like to be near water, because water is polar covalent. So remember, it's easy to remember to draw water, right? Because it kind of looks like a Mickey Mouse head. So this part of a water molecule has the positive charges. They're going to be attracted to the negative portion of the phosphate head. These pieces here, these are the fatty acids. This is the lipid, the oil. They have no charge. They are neutral. So because they're neutral, they have no affinity for water because water's charged. So things that are charged and are oppositely charged are going to attract to each other, call that polar. Things that do not have a charge, non-polar. That's what they're trying to do. They're trying to lead you to understanding, and this is coming in chapter 5 and chapter 7 about this particular molecule, which is called a phospholipid, and it is what forms the membranes of all cells. So this is why we are mostly made of water, and this is how other molecules are going to be able to move through this membrane, but it's a barrier, but it can let things pass. And it's because of this nature where it has two parts, where it's partly charged and partly non-charged, that it can do that. So I wouldn't worry too much about that. But like I said, I think they're trying to lead you to by giving you examples of why this is important.
Yeah, because I understood, like, you know, polar has a charge, non-polar doesn't have a charge. And even like the phospholipids I remember from biology in high school. But like on questions about it, I guess like I would think, oh, they repel each other, but the answer kept being that like polar groups attract one another, and I didn't understand why that was the answer and not the repulsion.
Oh, right, because they have different charges, right? So polar doesn't just mean negatively charged. Polar can mean positively charged too, right? So this is why that was the answer. Because water, even water by itself, is polar covalent. This side of the water molecule is negatively charged, and yes, that's repelled by this head, but these positively charged hydrogens are attracted. They're going to form hydrogen bonds with this phosphate head. Right? They're trying to get you to think about it as more fluid and not just rigid and stuck permanently in one conformation or one orientation, right? And they're trying to get you to think about it as being more, just more fluid and changeable.
Okay, thank you. I understand better now.
All right, you're welcome. Anybody else? Yes, no, maybe so. Alrighty then. All right, anybody that has specific questions, hang around. But those of you that are, I'm going to end the recording